There, my name is Michelle Pridgeon, and I am a student at Regis University's School of Physical Therapy. Today I will be discussing wrist flexor and extensor muscle recruitment during isometric, concentric, and eccentric contractions. The purpose of this research was to investigate muscle recruitment of wrist flexor and extensor muscles during different contraction types. To do this, my team and I used surface EMG, which is a common tool used by biomechanists to measure muscle recruitment. Prior studies have found that wrist position and contraction type will result in different EMG amplitude. More specifically, a study by Garner et al. Found that EMG amplitude for eccentric contractions tend to be less than concentric contractions because during lengthening contractions there are additional passive components such as the contractile protein titin, connective tissue, and tendons contributing to force production within the muscle. Given this prior background research, we expected to see greatest muscle recruitment during concentric contractions due to a lack of additional passive forces and due to the fact that gravity will be working against the direction of movement, which will impose a greater need for muscle recruitment to overcome that external force. Our 8 participants were selected on a volunteer basis from our biomechanics class and were chosen only if they could perform pain-free wrist extension and flexion and did not have sensitivity to rubbing alcohol or adhesive from the electrodes. Just to note, since we did have such a small sample size of all healthy individuals, this data cannot be generalized across the entire population. To begin data collection, we shaved the skin of the subjects and abraded it with rubbing alcohol wipes in order to minimize impedance. Electrodes were then placed over the wrist extensors and flexors of the subject's dominant hand, and the placement was confirmed by observing the EMG signal. The maximal voluntary contractions were determined using manual resistance. The subject performed isometric, concentric, and eccentric contractions for both the wrist extensor and flexor muscles while holding a 1-pound weight. After recording the peak EMG values and determining percent MVC values, mean and standard deviation of the percent MVC were calculated for the 3 contraction types. As can be seen in Figure 1, the blue bar represents percent MVC of flexors while the yellow bar represents percent MVC of extensors. The mean percent MVC values for wrist extensors were consistently higher across all contractions when compared to the percent MVC for wrist flexors. This observation may be due to weakness in the extensors. Also, another major takeaway from this graph is that concentric contractions produced the greatest percent MVC in both flexors and extensors. One last thing I'd like to note in regards to Figure 1 is that there were MVC values of over 100%, which indicates that a true MVC was not obtained. Based off of the results, our hypothesis was correct. Concentric contractions did cause the greatest muscle recruitment in both the wrist flexors and extensors. We expect this is due to the lack of passive forces from connective tissues and tendons because in all concentric conditions, the wrist was lifted up against the force of gravity. This research can be applied in the clinic when prescribing exercises to increase muscle recruitment in either the wrist flexors or extensors, which in this case we would suggest concentric contractions. It is also important for therapists to be aware that wrist position and contraction type plays a role in muscle recruitment. In conclusion, we found wrist extensors to be weaker than wrist flexors, which resulted in a higher percent MVC for wrist extensors, and concentric contractions caused the greatest muscle recruitment for wrist flexors and extensors. Thank you for listening.